This lecture covers nervous tissue. The nervous system is one of great complexity and mystery, and it'll absorb our attention in the next 5 chapters or so. But it is the foundation of all our conscious experience, our personality, and our behaviors. It profoundly intrigues biologists, physicians, psychologists, and even philosophers. The scientific study of the nervous system is called neurobiology. We can say that it combines both the life science, the biology, and the behavioral aspects of it as well. Throughout the lectures, when you see slides that are like this, that have a colored background, these are our learner objectives. They help to focus our learning for each section. In this section, our expected outcomes are to describe the overall function of the nervous system and to describe its major anatomical and functional subdivisions. If our bodies are to maintain homeostasis, then our trillions of cells need to work together. Luckily, we have 2 systems overseeing these processes. They are mainly the endocrine system and the nervous system, working together to maintain our internal coordination. The endocrine system communicates by means of chemical messengers, also known as hormones, that are secreted into the blood, and they target specific glands and organs within our body. The nervous system employs electrical and chemical means to send messages from cell to cell, to get our trillions of cells to work together. The nervous system carries out its tasks in 3 main basic steps. First of all, sense organs receive information about changes in the body and the external environment, and then transmit the coded messages to the brain and the spinal cord. The brain and the spinal cord are also known as the central nervous system, or CNS for short. We'll see that designation a lot. The central nervous system then processes the information and relates it to past experiences and determines what the appropriate response should be. And the central nervous system then issues commands to muscles and gland cells to carry out the correct response. Our nervous system has 2 major subdivisions. One is the central nervous system, or the CNS, which is the brain and the spinal cord. Both of those are so well protected in our body by our skull and our vertebral column. The other subdivision is the peripheral nervous system, also known as the PNS, and this is the rest of the nervous system that is not the brain or the spinal cord. It's composed of nerves and ganglia. A nerve is a bundle of nerve fibers, the axons, wrapped in fibrous connective tissues. And a ganglion, or ganglia is plural, is a knot-like swelling in a nerve where neuron cell bodies are concentrated. Again, these are part of the peripheral nervous system. Now the peripheral nervous system is divided again into further subdivisions called the sensory and the motor divisions. And then each of those themselves are divided even further into the somatic and the visceral subdivisions. So, the sensory division, which is also called the afferent division, carries signals from receptors to the central nervous system. If we take a look at the Latin of the word afferent, AF or af means toward, and fer means to carry. So think of the sensory division carrying signals and messages to the central nervous system to let our brain and our spinal cord know what's going on. So the sensory division, or afferent division, is then again divided into further categories known as the somatic sensory division, which carries signals from receptors in the skin, muscles, bones, and joints. to the central nervous system. And then also the visceral sensory division carries signals from the viscera, which are these deep organs like the heart, the lungs, and the stomach, and the urinary bladder. Those messages then are carried to the central nervous system. The motor division, also known as the efferent division, carries signals from the central nervous system to effectors, which are glands and muscles that can carry out a body response. Now let's take a look at the Latin here with the word efferent. EF means out or away from. And again, FER, fer, means to carry. So basically this is saying that this division carries messages away from the central nervous system. So again, this is divided into its two categories, just like the sensory division was. We have the somatic motor division, carries signals to the skeletal muscles, which then that output produces things like muscular contractions as well as reflexes, which are involuntary muscle contractions. Then the visceral motor division, which again has its own name, again the autonomic nervous system, carries signals to glands, cardiac, and smooth muscle. And these are involuntary responses that we cannot control, also known as visceral reflexes. So that division, the autonomic nervous system, or the visceral motor division, is divided even further yet into the sympathetic division and the parasympathetic division. The sympathetic division tends to arouse the body for action, like, for instance, accelerating heartbeat and increasing respiratory airflow. The sympathetic division does inhibit digestion, though. The parasympathetic division tends to have a calming effect. It slows heartbeat, but unlike the sympathetic division, which slows digestion, the parasympathetic division stimulates digestion. Here we have a diagram of the brain, the spinal cord showing the central nervous system, and then the rest showing the peripheral nervous systems. And this chart is very important. I want you to spend some time studying and memorizing this chart. I want you to be able to take a look at each of these and tell a little bit about each division and give examples. The terms that we've talked about can give the impression that we have several different nervous systems, but those are just terms of convenience. However, there is only one nervous system, and the subsystems that we've talked about here in this chart are interconnected parts that makes the whole nervous system. In our next section, we'll talk about the properties of neurons, and our expected outcomes or objectives are the following: to be able to describe 3 functional properties found in all neurons, to define the 3 most basic functional categories of neurons, to identify the parts of a neuron, and to explain how neurons transport materials between the cell body and tips of the axon. The role of communication in the nervous system is carried out by nerve cells, which are also called neurons. And these cells have 3 fundamental physiological properties that enable them to communicate with other cells. The first one is excitability, also called irritability. In other words, they're excitable. They can respond to environmental changes, like stimuli, and neurons exhibit this property to the highest degree. They exhibit conductivity. Neurons respond to stimuli by producing electrical signals that are quickly conducted to other cells, even at distant locations. And then secretion, number 3. When the signal reaches the end of a nerve fiber, the neuron secretes a neurotransmitter that crosses the gap and it stimulates the next cell. Over to the right, we actually see a picture of a neuron with its parts. Neurons are divided into 3 general classes, and you'll notice the names of these correspond to the 3 major aspects of the nervous system functions that were listed earlier. So, the first type is sensory or afferent neurons. These are specialized to detect stimuli like light, heat, pressure, and chemicals, and they transmit information about that to the central nervous system. Such neurons begin in almost every organ of the body and end in the central nervous system. And the word afferent again refers to signal conduction toward the central nervous system. Some receptors, such as those for pain and smell, are themselves neurons, but in some other cases, such as in the cases of taste and hearing, the receptor is actually a separate cell that communicates directly with the sensory neuron. Interneurons are also called association neurons, and they lie entirely within the central nervous system, and they connect the motor and sensory pathways. In fact, about 90% of all neurons are interneurons. The word interneuron refers to the fact that they lie between, and they interconnect the incoming sensory pathways and the outgoing motor pathways of the central nervous system. Motor neurons are also called efferent neurons. They send signals out to the muscles and the gland cells, which muscles and gland cells are also called the effectors. Remember, this means going away from the central nervous system. On this slide, we have a diagram of the 3 main classes of neurons to remind us how sensory neurons pick up information and send it to the central nervous system. Interneurons, or association neurons, are confined in the central nervous system themselves, but act to link the two. And then motor neurons send messages away from the central nervous system, also called efferent neurons, and then target muscles and glands, also called effectors. An important objective of the chapter is to learn the structure of a neuron. So we have a diagram off to the right of a typical motor neuron of the spinal cord. Though there are other types, this is a good representative. The soma, also known as the cell body commonly, is the control center of the neuron. That's right here. It has a single centrally located nucleus with a large nucleolus inside that. Then there's the cytoplasm around that nucleus, which contains items like the mitochondria. Remember, that's the powerhouse of the cell where ATP is made. Lysosomes, which contain digestive enzymes. The Golgi complex, which packages and exports cell materials. And other inclusions like the rough ER. Rough meaning it's an endoplasmic reticulum that contains ribosomes. ER is like little passageways within the cell, and the ribosomes that are located on there are the sites of protein synthesis. And then of course there's also the cytoskeleton. It consists of a dense mesh of microtubules and neurofibrils, which are bundles of actin filaments. The cytoskeleton compartmentalizes the rough ER into dark staining areas called Nissl bodies. These are named after Franz Nissl, who was a German neuropathologist. Other inclusions include glycogen, which is a storage form of glucose, lipid droplets, melanin, which is a pigment, and then lipofuscin pigment. This is produced when lysosomes digest old organelles, and as time builds up and the neuron gets older, this gets darker and darker, and more it actually can push the nucleus off to the side, which kind of lets us know that, oh, this is an older neuron here that we're dealing with. Neurons don't have any centrioles in their adult form, and therefore will not undergo any further mitosis. But even into old age, however, there's unspecialized stem cells in the central nervous system that can divide and develop into new neurons. But even considering, the neurons are unusually long-lived cells and can function for over 100 years in humans. The soma, or the cell body, usually gives rise to a few thick processes that branch into what's called a vast number of dendrites. These are named for resembling tree branches. In fact, that's what their name means, little tree branch. They're the primary site for receiving signals from other neurons, and the more dendrites a neuron has, the more information it can receive. Some neurons only have one dendrite, and some can have thousands. They provide a precise pathway for the reception and processing of information. On one side of the soma, or cell body, is a mound called the axon hillock, shown right here in the picture. From there, the axon, or nerve fiber, originates, shown here. The axon is cylindrical and relatively unbranched for most of the length, although along the way there may be some what are called axon collaterals, shown right here in the picture, that branch off. Most axons branch extensively at their distal end. An axon is specialized for rapid conduction of nerve signals to points remote from the soma. Its cytoplasm is called the axoplasm, and its cell membrane is the axolemma. A neuron never has more than one axon, and some neurons, like in the retina and the brain, have none. They may also be enclosed in a myelin sheath. The distal end of the axon has what's called terminal arborization. Here we can see the Latin meaning here, arbor, which has to do with trees, looking at like tree branches. This is an extensive complex of fine branches. We can see in the picture down here. And then there's synaptic knobs, or terminal buttons, which are little swellings at the end that form a junction, or the synapse, with the next cell. And these areas contain synaptic vesicles that are capable of making neurotransmitters. Some of these range in length from just a few micrometers to being up to a meter long. Neurons are categorized according to the number of processes that extend from the soma. One type, and actually the most popular and numerous type, are called multipolar neurons. This has one axon and multiple dendrites, and they're the most common, especially in the central nervous system. A bipolar neuron has one axon and one dendrite, common in olfactory cells, that's for smell, in the retina of the eye, and in the inner ear. Unipolar neurons are a single process leading away from a soma. A short distance away from the soma, the process branches like a T into a peripheral fiber and a central fiber. Peripheral fiber begins with a sensory ending, often far away from the soma. In the skin, for example, its signals travel toward the soma, but bypass it and then continue along the central fiber for the short remaining distance to the spinal cord. The dendrites are considered to be only the short receptive endings. The rest of the process, both peripheral and central, is the axon, defined by the presence of myelin and the ability to generate action potentials. Anaxonic neurons have many dendrites, but no axon. These are common in the retina of the eye, in the brain, and the adrenal gland. They are unable, usually, to generate action potentials. Many items, like proteins and organelles, need to be able to move along an axon, whether it be away from the soma or toward the soma. For instance, proteins may be needed to repair the axonema, or to serve as gated ion channels, or enzymes or neurotransmitters. This is called axonal transport. It's the two-way passage of proteins and organelles and other materials along an axon. And it can go in two different directions. Movement down the axon away from the soma is called anterograde transport, while movement up the axon toward the soma is called retrograde transport. Microtubules will guide these materials along the axon, and there's a couple different kinds that assist this. Motor proteins like kinesin and dynein carry materials on their backs, literally while they crawl along microtubules. Kinesin is motor proteins that move in anterograde transport, while the dynein are the motor proteins in the retrograde transport. Axonal transport is then divided into two different categories based on the speed. Fast axonal transport can move at a rate of 20 to 400 millimeters a day. Fast anterograde transport moves items like organelles and enzymes and small molecules. Fast retrograde transport is for recycled materials. It can return used synaptic vesicles and other materials back to the soma and inform the soma of conditions at the axon terminals. Unfortunately, some pathogens exploit this movement, such as the rabies virus, the herpes virus, tetanus, and polio viruses. The delay between infection and the onset of symptoms corresponds to the time needed for the pathogens to reach the somas. Slow axonal transport can only move a half to about 10 millimeters a day. You can think about it kind of like a train moving along tracks, that the train would move just as fast as the fast axonal transport, but in slow axonal transport it actually makes a lot of stops, which then slows things down. This is always anterograde, meaning it's moving away from the soma. It moves items like enzymes and cytoskeletal components. And also damaged nerve fibers will regenerate at a speed governed by slow axonal transport. In this section, we'll be looking at the supportive cells of the nervous system, also known as the neuroglia. Our expected learning outcomes or objectives are the following: to be able to name the 6 types of cells that aid neurons and state their respective functions. To describe the myelin sheath that is found around certain nerve fibers and explain its importance. To describe the relationship of unmyelinated nerve fibers to their supportive cells, and to explain how damaged nerve fibers can regenerate. Neuroglia are known as the supportive cells of the nervous system. There's about 1 trillion neurons in the nervous system, and that's 10 times as many neurons in your body as there's stars in our galaxy. Because they branch so extensively, they make up about 50% of the volume of the nervous system, but yet they're outnumbered at least 10 to 1 by the cells called the neuroglia, or also known as glial cells. Glial cells protect the neurons and help them function. The word glia means glue, and it actually implies their role to bind neurons together and provide a supportive framework for the nervous tissue. In the fetus, they form a scaffold that guides young migrating neurons to their destination. Wherever a mature neuron is not in synaptic contact with another cell, it's covered with glial cells. This prevents neurons from contacting each other except at points specialized for signal transmission, thus giving precision to their conduction pathways. There's 4 types of glia cells in the central nervous system and 2 types in the peripheral nervous system. Let's look at the 4 types in the central nervous system first. The first type is called oligodendrocytes. They form the myelin sheaths in the central nervous system that speed signal conduction. When I look at them, they remind me of an octopus because of the arms that extend out and then wrap around the nerve fibers to form the myelin sheaths. Ependymal cells line internal cavities of the brain, and it's their job to secrete and circulate cerebral spinal fluid, or CSF. They're cuboidal in shape, so they are termed cuboidal epithelium, though they don't have a basement membrane, and they have cilia on their apical surface. Microglia wander through the central nervous system looking for debris or for damage that they can help fix. Basically, they're big macrophages with a lot of extensions on them. They develop from white blood cells called monocytes and become concentrated in areas of damage. It's what doctors look for when they're looking for brain damage. It's actually thought that quite often during the day they do kind of a complete checkup on the brain tissue, which that's kind of reassuring. Then there's astrocytes. Looking at the list here, it looks like they have a lot of jobs to do. It's no wonder, because they are the most abundant glial cell in the central nervous system and constitute over 90% of the tissue in some areas of the brain. They cover the entire brain surface and most non-synaptic regions in the neurons and the gray matter of the central nervous system, and they're named for their many-branched, somewhat star-like shape. So here's a list of their jobs. They can support a supportive framework for the nervous tissue. They have extensions called perivascular feet, which contact the blood capillaries and stimulate them to form a tight, protective seal called the blood-brain barrier. They convert glucose to lactate to supply these nutrients to neurons. They secrete nerve growth factors. Those help to regulate nerve development. They communicate electrically with neurons and influence synaptic signaling between them. They regulate the composition of the tissue fluid. When neurons transmit signals, they release neurotransmitters and potassium ions, So astrocytes absorb these and prevent them from reaching excessive levels in the tissue fluid. When neurons are damaged, astrocytes form hardened scar tissue and fill up the space formerly occupied by the neurons. This process is called astrocytosis or sclerosis. Here we can see a diagram of some of these neuroglial cells in the central nervous system. Here we have the octopus-like oligodendrocytes. Here we have the ependymal cells. We have the macrophage-like, with lots of branches, microglia. And then the astrocytes with their so many functions. These two types of neuroglia occur in the peripheral nervous system. First of all, there's Schwann cells. They envelop nerve fibers in the peripheral nervous system, a lot like the oligodendrocytes do in the central nervous system. They wind repeatedly around a nerve fiber and produce the myelin sheath, and then they assist in regeneration of damaged fibers. There's also satellite cells that surround the neurosomas in the ganglia of the peripheral nervous system, and they provide electrical insulation around the soma and regulate the chemical environment of the neurons. The myelin sheath is a spiral layer of insulation around a nerve fiber formed by oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. Their composition is mostly like a plasma membrane, about 20% protein and 80% lipids. For the lipids, it includes phospholipids, glycolipids, and cholesterol. The process of forming the myelin around the neurons is called myelination. It actually begins at week 14 of fetal development and then proceeds very rapidly during infancy, which is why you see a lot of milestones and rapid growth in the first year of life of an infant, and it's completed in late adolescence. Due to the fact that these lipids are laid down in the brain and the nervous system during this time, it's important that a growing child gets dietary fat. Therefore, giving them skim milk would not be good. They should have the whole milk to be able to get the amount of fat that they need to myelinate their nerves. In the peripheral nervous system, Schwann cells spiral repeatedly around a single nerve fiber. They lay down as many as 100 layers of membrane with no cytoplasm between the membranes, then leaving the neurolemma, which is the thick outermost coil of the myelin sheath, on the outside, which will then contain the nucleus and most of the cytoplasm. And external to this neurolemma is the basal lamina, and a thin layer of fibrous connective tissue called the endoneurium. If you want to visualize this a little bit better, think about taking an almost empty tube of toothpaste and wrapping it tightly around a pencil. The pencil would represent the axon of the neuron, and then the spiral layers of the toothpaste tube would represent the myelin. The toothpaste that's left over in the tube would be forced to one end of the tube, which would then form a bulge on the external surface of the wrapping, which would then represent the body of a Schwann cell. Here's a diagram of what that may look like. And here again. In the central nervous system, an oligodendrocyte myelinates several nerve fibers in its immediate vicinity. It's like the octopus arms reaching out, and then it anchors to multiple nerve fibers. It cannot migrate around any one of them like a Schwann cell does, but it must push newer layers of myelin under the older ones, so the myelination therefore spirals inward toward the nerve fiber. And nerve fibers in the central nervous system have no neurolemma or endoneurium. And here's a diagram of what that might look like. Many Schwann cells or oligodendrocytes are needed to cover one nerve fiber. The myelin sheath is then segmented. So, within these segments, we have what's called the nodes of Ranvier. Many people may pronounce this Ranvier, but this guy that discovered them was French, so it's actually pronounced Ranvier. And these are the gaps between the segments. Then the internodes are the myelin-covered segments from one gap to the next. The initial segment is the short section of the nerve fiber between the axon hillock and the first glial cell. Since the axon hillock and the initial segment play an important role in initiating a nerve signal, they're collectively called the trigger zone. So how about glial cells and brain tumors? A tumor consists of a mass of rapidly dividing cells, but we need to remember that mature neurons, however, have little or no capacity for mitosis and seldom form tumors because of that. Basically, a tumor is a cell who keeps undergoing mitosis again and again and again without any control. So, most brain tumors arise from either the meninges, which are the protective membranes of the central nervous system, from metastasis, cells that are cancerous coming in, traveling to the area from other sites, such as in colon cancer, or from glial cells that are mitotically active throughout the life. Therefore, these cancers of the glial cells are called gliomas. They grow rapidly and are very highly malignant. Because of the blood-brain barrier, it decreases the effectiveness of chemotherapy. Often these must be treated by radiation or by surgery. There are some degenerative disorders or diseases of the myelin sheath that can be found in humans. One of those such diseases is called multiple sclerosis. This is when the oligodendrocytes and the myelin sheaths in the central nervous system deteriorate and are replaced by hardened scar tissue. This disrupts nerve conduction and can cause symptoms such as double vision and tremors, numbness, and speech defects, and usually onsets between the ages of 20 to 40. And can be fatal within from the first year all the way to 25 to 30 years. Scientists are unsure of the cause of this even still today, but have hypothesized that it could be an autoimmune system disease triggered by a virus to genetically susceptible people. Another degenerative disorder of the myelin sheath is called Tay-Sachs disease. It's a hereditary disorder of infants found quite often in individuals of Eastern European Jewish ancestry. It's what's called a homozygous recessive disorder, meaning an individual who has two recessive alleles would get this disorder. It's caused by the abnormal accumulation of a glycolipid called GM2 in the myelin sheath, which normally, when it's made, would be decomposed by a lysosomal enzyme. But the enzyme is missing in individuals that are homozygous recessive for Tay-Sachs. So, GM2, also called ganglioside, accumulates and then disrupts the conduction of the nerve signals, resulting in blindness, loss of coordination, dementia, and then usually fatal around age 3 to 4. Symptoms can many times be noticed around age 1. There's many central nervous system and peripheral nervous system fibers that are not myelated or unmyelinated. In the peripheral nervous system, the Schwann cell holds 1 to 12 small fibers in subsurface grooves that we can see in the picture here. And then the membrane folds around each fiber. And then the outer covering here is the basal lamina. Conduction speed of nerve fibers. The speed at which a nerve signal travels along the surface of a nerve fiber depends on two factors. Number one, the diameter of the fiber. Large fibers will have more surface area and therefore will conduct signals much more rapidly. And then also on the presence or absence of myelin. Myelin can speed up signal conduction. Small unmyelinated fibers can travel 1/2 to 2 meters per second. Small myelinated fibers can go from 3 to 15 meters per second, and large myelinated fibers up to 120 meters per second. One might ask, then why don't we have all large myelinated fibers? Well, the answer to that would be there's just not enough room for that, and besides, sometimes we don't need such speed. Slow signals to the gastrointestinal tract are just fine. Fast signals to the skeletal muscles, where speed improves balance and coordinated body movement, then is necessary. Nerve fibers of the peripheral nervous system are vulnerable to cuts, crushing injuries, and other trauma. A damaged peripheral nerve fiber can regenerate, however, if its soma is intact and at least some of the neurolemma remains. In the steps of regeneration, when a nerve fiber is cut, the fiber distal to the injury cannot survive because it's incapable of protein synthesis. The protein synthesizing organelles are mostly in the soma. So as the distal fiber degenerates, so do its Schwann cells, which would depend on it for their maintenance. Macrophages clean up the tissue debris at the point of injury and beyond. The soma then exhibits a number of abnormalities on its own, probably because it's cut off from the supply of nerve growth factors from the neuron's target cells. The soma swells and the endoplasmic reticulum breaks up, so the Nissl bodies disperse and the nucleus moves off center. Not all damaged neurons can survive this. Some will die at this stage, but often the axon stump can sprout multiple growth processes. Muscle fibers that are deprived of their nerve supply at this point will exhibit a shrinkage called denervation atrophy. Near the injury, Schwann cells, the basal lamina, and the neurolemma form what's called a regeneration tube. The Schwann cells produce cell adhesion molecules and nerve growth factors that enable a neuron to regrow to its original destination. When one growth process finds its way into the tube, it grows rapidly, like 3 to 5 millimeters a day, and the other growth processes are retracted. The regeneration tube guides the growing sprout back to the original target cells, reestablishing synaptic contact. And when contact is established, the soma shrinks and returns to original appearance, and the re-innervated muscle fibers can regrow. Regeneration is not fast. It can take up to 2 years, And it's not always perfect, and it's not always possible. In fact, some nerve fibers might even connect with the wrong muscle fibers, and some will eventually die anyway. Regeneration of damaged nerve fibers in the central nervous system cannot occur at all, but because of the extra protection of the skull, they're not damaged as often. Here's a diagram showing all the stages of regeneration of a motor nerve fiber. Be sure to study those and study the steps involved. Nerve growth factor is a protein secreted by a gland, a muscle, or glial cells, and then picked up by axon terminals of neurons. It prevents the cell death, or apoptosis, in growing neurons. And it enables growing neurons to make contacts with their targets. It was isolated by Rita Levi-Montalcini in the 1950s. She won a Nobel Prize in 1986 with Stanley Cohn. The use of growth factors is now a vibrant field of research. In this section, we'll look at the electrophysiology of neurons. Our objectives are to explain why a cell has an electrical charge difference, or a voltage, across its membrane, to explain how stimulation of a neuron causes a local electrical response in its membrane, to explain how local responses generate a nerve signal, and explain how the nerve signal is conducted down an axon. The nervous system has intrigued scientists and philosophers since ancient times. The Roman physician Galen thought that the brain pumped a vapor called psychic pneuma through hollow nerves and squirted it into the muscles to make them contract. The French philosopher René Descartes in the 17th century still supported Galen's theory, but it finally fell out of favor in the 18th century. This is when Luigi Galvani discovered the role of electricity in muscle contraction. An Italian histologist named Camillo Golgi, that last name should sound familiar to you, as in Golgi bodies of the cell, he developed an important method for staining neurons with silver. This was in the 19th century. This technique enabled a Spanish histologist, Santiago Ramon y Cajal, I hope I said that right, with, of course, tremendous skill and patience, to trace the course of nerve fibers through tissue sections. He demonstrated that the nervous pathway was not a continuous wire or a tube, but a series of cells separated by the gaps that we now call synapses today. Golgi and Cajal shared the 1906 Nobel Prize for Physiology Medicine for these important discoveries. Cajal's theory brought up two very important key questions. Number one, how does a neuron generate an electrical signal? And how does it transmit a meaningful message to the next cell? Which this section will answer. Electrophysiology is the study of cellular mechanisms for producing electrical potentials or currents. It's the basis for neural communication and muscle contraction. Neural communication, like muscle excitation, is based on electrophysiology, cellular mechanisms for producing electrical potentials and currents. An electrical potential is a difference in the concentration of charged particles between one point and another. For instance, living cells are polarized and have a resting membrane potential. Cells actually have more negative particles on the inside of their membrane than on the outside. Neurons have about a negative 70 millivolt resting membrane potential. This resting membrane potential means that our cells are polarized. It means there's a charge difference across the plasma membrane. Typically, again, it's about negative 70 millivolts in an unstimulated resting neuron. And that negative value once again means that there's more negatively charged particles on the inside of the membrane than on the outside. We don't have free electrons in the body as we do in an electrical circuit. Electrical currents in the body are created instead by the flow of ions, such as sodium and potassium, Na and K, through channels in the plasma membrane. These are gated channels. Gated channels can be opened and closed by various stimuli, which enable cells to turn electrical currents on and off. The reason a cell has a resting membrane potential is that the electrolytes are unequally distributed between the extracellular fluid on the outside of the plasma membrane and the intracellular fluid on the inside. The resting membrane potential results from the combined effect of three factors: the diffusion of ions down their concentration gradient through the membrane, the selective permeability of the membrane, allowing some ions to pass more easily than others, and number 3, the electrical attraction of cations and anions to one another. Potassium, or K+, has the greatest influence on the resting membrane potential because the plasma membrane is actually more permeable to potassium than any other ion. Imagine a hypothetical cell in which all the potassium starts out in the intracellular fluid, with none in the extracellular fluid. Also in the intracellular fluid, there's a number of other cytoplasmic anions that cannot escape from the cell because of their size or charge. These would be things like phosphates, sulfates, other small organic acids, proteins, ATP, and RNA. Assuming that potassium can diffuse freely through the channels in the plasma membrane, it diffuses down its concentration gradient and out of the cell, leaving these cytoplasmic anions behind, and as a result, the intracellular fluid grows more and more negatively charged. But as the intracellular fluid becomes more negative, it exerts a stronger attraction for the positive potassium ions outside of the cell and then attracts some of them back. Eventually an equilibrium is reached in which potassium moving out of the cell, down its concentration gradient, and then into the cell by electrical attraction at equal rates. There's no further net diffusion of potassium. At this point of equilibrium, potassium is about 40 times as concentrated in the intracellular fluid as in the extracellular fluid. If potassium was the only ion affecting resting membrane potential, it would give the membrane a potential of about negative 90 millivolts. However, sodium ions also enter the picture. Sodium symbol is Na. Sodium is about 12 times as concentrated on the extracellular fluid as in the intracellular fluid, so that would be opposite of potassium. The resting plasma membrane is much less permeable to sodium than to potassium, but sodium does diffuse down its concentration gradient into the cell, attracted by the negative charge in the intracellular fluid. This sodium leak is only a trickle, but it's enough to cancel some of the negative charge and reduce the voltage across the membrane. Sodium leaks into the cell and potassium leaks out, but the sodium-potassium pump continually compensates for this leakage. It moves about 3 sodium out for every 2 potassium that it brings in, and this actually consumes 1 ATP for each exchange cycle. By removing more cations from the cell than it brings in, it contributes about -3 millivolts to the resting membrane potential. The net effect of all this potassium diffusion out of the cell and sodium diffusion inward, and the sodium-potassium pump, leaves about a resting membrane potential of about -70 millivolts. The sodium-potassium pump accounts for about 70% of the energy, or ATP, requirement of the nervous system. Every signal generated by a neuron slightly upsets the distribution of sodium and potassium, so the pump must work continually to restore that equilibrium. And that's why nervous tissue has one of the highest rates of ATP consumption of any tissue in the body, and why it demands so much glucose and oxygen. The uneven distribution of sodium-potassium on the two sides of the plasma membrane pertains only to a very thin film of ions immediately adjacent to the membrane surfaces, not the ones further away. Here's a diagram of the sodium-potassium pump. Be sure to study that diagram and that section of your textbook, and visualize that in your mind of how that works. Stimulation of a neuron causes local disturbances in the membrane potential. Typically, but with exceptions, the response will begin at a dendrite, spread through the soma, and then travel down the axon, and then end at the synaptic knobs. We'll consider the process in that order. A local potential is a change in the membrane potential of a neuron occurring at and nearby the part of the cell that's stimulated. Different neurons can be stimulated by chemicals, light, heat, or even mechanically. A chemical stimulate, we'll say that as our example, binds to a receptor on the neuron. This opens sodium gates and then allows sodium to enter the cell. Remember, sodium is a positive ion, so the entry of these positive ions makes the cell less negative. This is called depolarization. Anytime voltage shifts to a less negative value, again, that's called depolarization. A change in membrane potential towards 0 millivolts occurs when these sodium gates open. The sodium entry results in a current that travels toward the cell's trigger zone. This short-range change in voltage is called a local potential. There are 4 characteristics that distinguish local potentials from action potentials, which we'll study in a little bit. Local potentials are graded, meaning they vary in magnitude with stimulus strength. For instance, a stronger stimulus can open more sodium gates. They're decremental, meaning they get weaker the further they spread from the point of stimulation. They're reversible. If stimulation ceases, the cell quickly returns to its normal resting potential. And they can be either excitatory or inhibitory. For instance, some neurotransmitters actually make the membrane potential more negative or hyperpolarize it, so it becomes less likely to produce an action potential. An example of a neurotransmitter that does that is called glycine. Here's a diagram showing local potentials. An action potential is a dramatic change in membrane polarity produced by voltage-gated ion channels. It only occurs where there's a high enough density of voltage-regulated gates that are opened up. In the soma, there's only about 50 to 75 gates per micrometer squared, and that cannot generate an action potential alone. But down by the trigger zone, there's 350 to 500 gates per micrometer squared. And this is usually where the action potential is generated, if it can reach all the way down there. If excitatory local potential reaches this trigger zone and is still strong enough, it can open these gates and generate an action potential. It's a rapid up and down shift in the membrane voltage involving a sequence of steps. First of all, there's the arrival of current at the axon hillock that depolarizes the membrane. Remember that depolarization is when there's a voltage shift to a less negative value. Depolarization must reach a threshold, which is a critical voltage. It's about negative 55 millivolts, and that's the amount that's required to open the voltage regulated gates. Voltage gated sodium channels then open, and sodium enters and depolarizes the cell, which then opens more channels, resulting in a rapid positive feedback cycle as voltage rises. The neuron now fires, or produces an action potential. At threshold, voltage-gated sodium channels open quickly, while potassium channels open more slowly. The initial effect on membrane potential is therefore due to sodium. Initially, only a few sodium channels open, but as, of course, sodium enters the cell, it creates that positive feedback loop. As the rising potential passes 0 millivolts, sodium channels are inactivated and begin closing. By the time they all close and sodium inflow ceases, the voltage peaks at approximately about positive 35 millivolts, sometimes as low as 0, sometimes as high as 50. The membrane is now positive on the inside and negative on the outside. Its polarity has been reversed compared to resting membrane potential. By the time the voltage peaks, the slow potassium channels are fully open, finally. Potassium ions, repelled by the positive intracellular fluid, now exit the cell. Their outflow repolarizes the membrane. That is, it shifts the voltage back into the negative numbers. The action potential consists of this up and down voltage shift that occurs from the time the threshold is reached to the time the voltage returns to resting membrane potential. Potassium channels stay open longer than sodium channels, so slightly more potassium leaves the cell than the amount of sodium that entered. Therefore, the membrane voltage drops to 1 or 2 millivolts, more negative than the original resting membrane potential, producing a negative overshoot called hyperpolarization, or after potential. As you can see, sodium and potassium switch places across the membrane during an action potential. During hyperpolarization, sodium diffusion into the cell, and in the central nervous system, the removal of the extracellular potassium by astrocytes gradually restores the original resting membrane potential. Only a thin layer of the cytoplasm next to the cell membrane is affected. In reality, very few ions are involved in this. The action potential is often called a spike, as it happens so fast. Some of these diagrams are deliberately distorted so that we can actually visualize this a little bit better. Here's a diagram to help us visualize these gated channels and the action potential that occurs. We've talked about the characteristics of a local potential. Here's the characteristics of action potentials. It follows an all or none law. If a stimulus depolarizes the neuron to threshold, the neuron fires at its maximum voltage, such as positive 35 millivolts. If threshold is not reached, it doesn't fire. Above threshold, stronger stimuli don't produce stronger action potentials. Thus, action potentials are not graded. They're non-decremental. They don't get weaker with distance. They're irreversible. Once started, it goes to completion and cannot be stopped. Make sure you check out the diagrams contrasting action and local potential. During an action potential and for a few milliseconds after, it can be difficult or even impossible to stimulate that same region of the neuron to fire again. That period is called the refractory period. It's the period of resistance to stimulation. It can be two different phases. One is an absolute refractory period, in which no stimulus of any strength will trigger a new action potential. And this lasts as long as those sodium gates are open and then inactivated. Then there's a relative refractory period. It's only especially strong stimuli can trigger a new action potential. During this time, the potassium gates are still open, and any effect of incoming sodium is opposed by the outgoing potassium. And this generally lasts until hyperpolarization ends. Remember, this is only on a small patch of a neuron's membrane, not the whole membrane. Other parts can still be stimulated. Now let's take a look at signal conduction in whole nerve fibers. Unmyelinated fibers have voltage-gated channels along their entire length. Action potential at the trigger zone causes sodium to enter the axon and diffuse into adjacent regions. This depolarization excites voltage-gated channels. An opening of the voltage-gated ion channel results in a new action potential, which then allows sodium diffusion to excite the membrane immediately distal to that. And it acts like a chain reaction that continues until the nerve signal reaches the end of the axon. Kind of like a nerve signal, like a wave of falling dominoes. Each one triggers the next in line. One might think, well, could it move backwards? And the answer is no. Because of the refractory period, directly behind where the new ones are being generated, it keeps things moving in one direction. Generally from the soma on down the axon. Myelinated fibers conduct signals slightly different from the unmyelinated ones. Myelinated fibers conduct signals with saltatory conduction. It's a signal that seems to jump from node to node. The nodes of Ranvier contain many voltage-gated ion channels, while the myelin-covered inner nodes contain few. When sodium enters the cell at a node, its electrical field repels positive ions inside the cell. As these positive ions move away, their positive charge repels their positive neighbors, transferring energy down the axon rapidly, conducting the signal. Myelin speeds up this conduction by minimizing leakage of sodium out of the cell and further separating the inner positive ions from attraction of negative ions outside the cell. But the signal strength does start to fade in the internode. But luckily, when the signal reaches the next node of Ranvier, it's strong enough to open more voltage-gated ion channels and add a new full-strength action potential. Again, this is called saltatory conduction. You can think of it like an analogy to a crowded subway car. The doors open, which would be like the sodium gates at a node. 20 or more people get on, like sodium flowing into the axon, and then everyone has to push to the rear of the car to make room for them. No one passenger moves from the door to the rear, but the crowding and the transfer of energy from person to person forces even those at the rear to move a little, like the sodium ions at the next node. Events at one node thus create excitation at the next node even some distance away. Here's a diagram depicting that. Be sure to study these diagrams in the textbook. It's much faster than the conduction in unmyelinated fibers. In a myelinated fiber, it can be up to 120 meters per second, but in the unmyelinated ones, it's only up to 2 meters per second. In this section, we'll take a look at synapses. Our objectives are to explain how messages are transmitted from one neuron to another, and to give examples of neurotransmitters and neuromodulators and describe their actions, and to explain how stimulation of a postsynaptic cell is stopped. A nerve signal soon reaches the end of an axon and it can go no further. So what happens? In most cases, it triggers the release of a neurotransmitter that stimulates a new wave of electrical activity at the next cell across the synapse. Here's how we describe the synapse between two neurons. The first neuron in the signal path is called the presynaptic neuron, and it's the one that releases the neurotransmitter. The second neuron is the postsynaptic neuron, and it responds to the neurotransmitter. A presynaptic neuron can synapse with a dendrite, a soma, or an axon of a postsynaptic neuron to form either an axodendritic, axosomatic, or axoaxonic synapses. A neuron can have an enormous number of synapses, like for instance spinal motor neurons are covered by about 10,000 synaptic knobs from other neurons, 8,000 ending in its dendrites, 2,000 in its soma, and in a region called the cerebellum of the brain, one neuron can have as many as 100,000 synapses. The synaptic cleft, which is the gap between neurons, was discovered by Ramon y Cajal through histological observations. Otto Loewy in 1921 demonstrated that neurons communicate by releasing chemicals, or call them chemical synapses. He flooded exposed hearts of two frogs with saline solution, stimulated their vagus nerve, the first frog, and the heart actually slowed. Then he removed saline from that frog and found it slowed the heart of the second frog. He named it Vagustaf, or vagus substance. Later, it's been renamed acetylcholine, and it was known as the first known neurotransmitter. Electrical synapses do exist. They occur between some neurons, some neuroglia, and cardiac and single unit smooth muscle. Between gap junctions that adjoin adjacent cells, the ions diffuse through the gap junctions from one cell to the next. It's an advantage of a quick transmission. There's no delay to actually release and bind the neurotransmitter. But the disadvantage is they cannot integrate information and make decisions. This ability is reserved for chemical synapses, in which neurons communicate with neurotransmitters. The structure of a chemical synapse. The synaptic knob of a presynaptic neuron contains synaptic vesicles that contain neurotransmitter. Many vesicles are docked on release sites on the plasma membrane and are ready to release the neurotransmitter. A reserve pool of synaptic vesicles is located further away from the membrane. The postsynaptic neuron membrane contains proteins that function as receptors and ligand-regulated ion gates. Here's an awesome picture showing those. Presynaptic neurons have synaptic vesicles with neurotransmitter, and postsynaptic have receptors and ligand-regulated ion channels, as shown in this diagram here. Neurotransmitters are molecules that are released when a signal reaches a synaptic knob that binds to a receptor on another cell and alters that cell's physiology. More than 100 neurotransmitters have been identified, but most fall into four major categories: acetylcholine, amino acids, monoamines, and neuropeptides. Acetylcholine is in a class by itself. It's formed from acetic acid and choline. Amino acid transmitters include glycine, glutamate, aspartate, aminobutyric acid, which is also nicknamed GABA or GABA. Monoamines are synthesized from amino acids by removing the carboxyl group while retaining the amino group. They include catecholamines like epinephrine, norepinephrine, and dopamine, and also include histamine, ATP, and serotonin. Neuropeptides contain 2 to 40 amino acids in the chain and are stored in secretory granules. They include cholecystokinin and substance P. Here's some diagrams of these neurotransmitters and related messengers. Synapses vary. Some are excitatory and others are inhibitory. Sometimes a transmitter's effect differs depending on the type of receptor and the postsynaptic cell. Some receptors are ligand-gated ion channels and others act through second messengers. Next, we'll consider 3 kinds of synapses. Cholinergic synapse uses acetylcholine, sometimes known as ACH for short. A nerve signal arrives at the synaptic knob and opens voltage-gated calcium channels. The calcium enters the knob and triggers exocytosis of acetylcholine. The acetylcholine diffuses across the cleft and binds to the postsynaptic receptors. The receptors are ion channels that open and allow sodium and potassium to diffuse. The entry of sodium causes a depolarizing postsynaptic potential. If depolarization is strong enough, it'll cause an action potential at the trigger zone. An inhibitory GABAergic synapse is one that employs aminobutyric acid as its neurotransmitter. The nerve signal triggers the release of GABA into synaptic clefts. GABA receptors are chloride channels. The chloride enters the cell and makes the inside more negative than the resting membrane potential. The postsynaptic neuron is inhibited and then less likely to fire. Adrenergic synapse employs the neurotransmitter norepinephrine, NE for short, also called noradrenaline. Norepinephrine and other monoamines and neuropeptides act through second messenger systems, such as cyclic AMP. The receptor is not an ion gate, but a transmembrane protein associated with the G protein. Slower to respond than cholinergic and GABAergic synapses, and has an advantage of enzyme amplification. A single molecule of norepinephrine can produce vast numbers of product molecules in the cell. cessation of the signal. The synapse must turn off stimulation to keep postsynaptic neuron from firing indefinitely. So the presynaptic cell stops releasing the neurotransmitter. And the neurotransmitter only stays bound to its receptor for about 1 millisecond, then it's cleared. The neurotransmitter diffuses into nearby extracellular fluid, and then our astrocytes come into play in the central nervous system and absorb it and return it to the neurons. The synaptic knob reabsorbs neurotransmitter by endocytosis as well. Like monoamine transmitters are broken down after reabsorption by monoamine oxidase. And acetylcholine is broken down by acetylcholinesterase in the synaptic cleft. After degradation, the presynaptic cell reabsorbs the fragments of the molecule for recycling. Neuromodulators are chemicals secreted by neurons that have long-term effects on groups of neurons. They can alter the rate of neurotransmitter synthesis and release and reuptake and breakdown. They may adjust the sensitivity of postsynaptic membrane. Nitric oxide is an example of a neuromodulator. It's a gas that enters the postsynaptic cells and activates second messenger pathways. For example, relaxing smooth muscle. Neuropeptides are chains of amino acids that act as neuromodulators. For instance, endorphins are neuropeptides that can inhibit pain signals in the central nervous system. In this section, we'll look at neural integration. Our objectives are the following: to explain how a neuron decides whether or not to generate action potentials, to explain how the nervous system translates complex information into a simple code, to explain how neurons work together in groups to process information and produce effective output, and to describe how memory works at the cellular and molecular levels. Neural integration is our ability to process, store, and recall information and to use it to make decisions. The chemical synapses allow for this decision making. And brain cells are incredibly well connected, allowing for complex integration. For instance, pyramidal cells of the cerebral cortex have about 40,000 contacts with other neurons. And there's a trade-off. Chemical transmission involves a synaptic delay that makes information travel slower than it would be if there was no synapse. Neural integration is based on the postsynaptic potentials produced by neurotransmitters. So remember that a typical neuron has a resting membrane potential of about -70 millivolts, and that a threshold of about -55 millivolts. A neuron has to be depolarized to this threshold in order to produce an action potential. Any voltage change in that direction makes a neuron more likely to fire, and therefore it's called an excitatory postsynaptic potential, or an EPSP. Usually they result from sodium flowing into a cell and then neutralizing some of the negative charge on the inside of the membrane. In other cases, a neurotransmitter hyperpolarizes the postsynaptic cell and makes it more negative than the resting membrane potential. Since this makes the postsynaptic cell less likely to fire, it's called an inhibitory postsynaptic potential, or an IPSP. Some gated chloride channels causing chlorine to flow into the cell. Chlorine, remember, is a negative ion, and then make the cytosol more negative. A less common way to open selective potassium channels is increasing potassium diffusion out of the cell. Different neurotransmitters can cause different types of postsynaptic potentials in the cells that they bind to. For instance, glutamate and aspartate produce EPSPs in brain cells, and glycine and GABA produce the opposite. A neurotransmitter might excite some cells and inhibit others, depending on the type of receptors that the postsynaptic cells have. For instance, acetylcholine and norepinephrine work in this way. Acetylcholine excites skeletal muscle but inhibits cardiac muscle because of the different receptors involved. One neuron can receive input from thousands of other neurons. Some of the incoming nerve fibers can produce EPSPs while others produce IPSPs. The neuron's response depends on whether the net input is excitatory or inhibitory. If EPSPs override the IPSPs, threshold may be reached and set off an action potential, but in reverse, it could inhibit the neuron from firing. Summation is the process of adding up postsynaptic potentials and responding to their net effect. It occurs in the trigger zone. It's fundamentally a balance between EPSPs and IPSPs that enables the nervous system to make decisions. It's kind of like a majority vote or a cellular democracy. There's two ways in which EPSPs can add up to do this, and they can actually occur simultaneously. In temporal summation, it occurs when a single synapse generates EPSPs so quickly that each is generated before the previous one fades, and this can add up over time to hit the threshold voltage that triggers an action potential. In spatial summation, this occurs when EPSPs from several different synapses add up to the threshold at the axon hillock. Several synapses admit enough sodium to reach the threshold, and the neuron can fire. This is an example of facilitation, which is a process in which one neuron enhances the effect of another. In presynaptic inhibition, it's a process in which one presynaptic neuron suppresses the other. It's the opposite of facilitation. It reduces or halts unwanted synaptic transmissions, like the inhibiting neuron, cell I in the figure below, releases GABA, and it prevents the voltage-gated calcium channels in the synaptic knob, S in the figure below, from opening, and so the knob releases little or no transmitter, and therefore no action potential. The nervous system must interpret and pass along both quantitative and qualitative information about its environment. For instance, is the light red or green? Is the taste salty or sour? Is the sound loud or soft? Considering the complexity of information to be communicated about conditions in and around the body, it seems a marvel that this can be done. But when we consider how we have our genetic code in a simple language of A, T, C's, and G's, we have a complex message in simple code with our nervous system as well. The way in which the nervous system converts information to a meaningful pattern of action potentials is called neural coding, or sensory coding, when it occurs in the sense organs. This is an example of neural coding. This figure is based on recordings made from sensory fibers in a frog's sciatic nerve, and as the gastrocnemius muscle was stretched by suspending weights from it. As the stimulus strength and stretch increase, the firing frequency of the neuron increases. Firing frequency is a coded message that informs the central nervous system of stimulus intensity. So far, we've dealt with interactions involving only a few neurons at a time, but actually neurons function in larger ensembles called neural pools. each of which consists of thousands to millions of interneurons concerned with a particular body function, such as the rhythm of your breathing, or moving limbs rhythmically when walking. Information arrives at a neural pool through one or more input neurons, which branch repeatedly and synapse with numerous interneurons in the pool. Some input neurons form multiple synapses with single postsynaptic cells. They can produce EPSPs at all points of contact with that cell, and through spatial summation make it fire more easily than if they were synapsed with it at only one point. Within the discharge zone an input neuron, that neuron acting alone can make the postsynaptic cells fire. But in a broader facilitated zone, it synapses with still other neurons in the pool with fewer synapses on each of them, and it can stimulate those neurons to fire only with the assistance of other input neurons. That is, it facilitates the others, like it has a vote on what the postsynaptic cells in the facilitated zone will do. But it cannot determine the outcome alone. Such arrangements, repeated thousands of times throughout the central nervous system, give neural pools great flexibility in integrating input from several sources and deciding on the appropriate output. The functioning of a radio can be understood from a circuit diagram by showing its components and their connections, and so similarly the functions of a neural pool are partly determined by its neural circuit, or the pathways among its neurons. There's four principal kinds of neural circuits. In a diverging circuit, one nerve fiber branches and synapses with several postsynaptic cells, and one neuron can produce an output through hundreds of other neurons. In a converging circuit, input from many nerve fibers can be funneled to one neuron or one neural pool. It's the opposite of a diverging circuit. In a reverberating circuit, neurons stimulate each other in a linear sequence, but one or more of the later cells re-stimulates the first cell to start the process all over, like for instance in the diaphragm and in your intercostal muscles. And in a parallel after discharge circuit, the input neuron diverges to stimulate several chains of neurons. Each chain has a different number of synapses. Eventually, they reconverge on one or a few output neurons, but with varying delays. After discharge, continued firing after the stimulus stops. Here's some diagrams to visualize each of those: the diverging, the converging. In diverging, one to many; converging is many to one. The reverberating, being able to send a message back to the beginning, and then the parallel after discharge. Here we'll talk about memory, such as you may be wondering, how am I going to remember all of this stuff that I'm learning in this textbook? The things we learn and remember are not stored in individual memory cells in the brain. We don't have a neuron assigned to remember our phone number and another assigned to remember our grandmother's face, for example. Instead, the physical basis of memory is a pathway through the brain called a memory trace, or an engram, in which new synapses have formed or existing synapses have been modified to make the transmission easier. In other words, synapses are not fixed for life. They change in response to our experiences. They can be added, taken away, and modified to make transmission easier or harder. Synapses can be created or deleted in as little as 1 to 2 hours. The ability of synapses to change is called synaptic plasticity. The process of making transmission easier is called synaptic potentiation. It's one form of synaptic plasticity. Neuroscientists still argue about how to classify our various forms of memory, but three kinds are generally recognized: immediate memory, short-term memory, and long-term memory, and each of these correlate with different modes of synaptic potentiation. Your immediate memory is the ability to hold something in your thoughts for a few seconds. It's essential for reading ability. Immediate memory may be based on reverberating circuits. Our impression of what just happened can thus re-echo in our minds for a few seconds as we experience the present moment and anticipate the next one. Our short-term memory, or STM, lasts from seconds to hours. It includes a working memory for taking action, such as remembering a phone number you just looked up. It includes the storage and appears to occur in circuits of facilitated synapses. It can be induced by tetanic stimulation, the rapid arrival of repetitive signals at a synapse. Each signal causes a certain amount of calcium to enter the synaptic knob. If the signals arrive very rapidly, the neuron cannot pump out all the calcium emitted by one action potential before the next action potential occurs. More and more calcium accumulates in the knob. Since calcium is what triggers the release of neurotransmitter, each new signal releases more neurotransmitter than the one before. With more neurotransmitter, the EPSPs in the postsynaptic cell become stronger and stronger, and that cell is more likely to fire. Memories lasting for a few hours, such as remembering something that someone said to you earlier in the day, can involve post-tetanic potentiation. In this process, the calcium level in the synaptic knob stays elevated. Therefore, little stimulation is needed to recover the memory. If an exceptionally large burst of neurotransmitter occurs, then that synapse is heavily used, and therefore a new stimulus can excite the postsynaptic cell more easily next time. Thus, your memory may need only a slight jog to recall something from several hours earlier. Long-term memory lasts up to a lifetime and is less limited than short-term memory in the amount of information it can store. Long-term memory allows you to memorize the lines of a play, or one hopes the information of a textbook for an exam. On a larger scale, it enables you to remember things about yourself, like how to get home and your childhood experiences. There's two forms of long-term memory. Declarative memory is the retention of events and facts that you can put into words, like numbers and names and dates. And procedural memory is the retention of motor skills, like how to tie your shoes, or how to play a musical instrument, or how to type on a keyboard. Some long-term memory involves molecular changes such as long-term potentiation, and some actually involves remodeling of synapses or the formation of new ones, such as the new branching of axons and dendrites. Long-term potentiation involves NMDA receptors. That means N-methyl D-aspartate, which is a chemical similar to glutamate. And it involves the NMDA receptors on dendritic spines of the pyramidal neurons. When NMDA receptors bind glutamate and receive tetanic stimuli, they allow calcium to enter the cell. And then calcium acts as a second messenger, causing more NMDA receptors to be produced, and the synthesis of proteins involved in synapse remodeling. And it releases signals like nitric oxide that trigger more neurotransmitter release from the presynaptic neuron. So speaking of memory, there are some disorders that degenerate memory. The first one is Alzheimer's disease. It involves about 100,000 deaths per year. It affects 11% of the population over 65 and 47% by age 85. Memory loss, moodiness, being combative, and losing the ability to walk, talk, and eat are all possible symptoms. People who have Alzheimer's show deficiencies in acetylcholine and nerve growth factor, and the actual disease can be confirmed upon autopsy by looking at the atrophy of the brain. As can be seen in the picture here, these parts have gotten smaller with the shrunken gyri and the wide spaces in between called the sulci. Parkinson's disease is a progressive loss of motor function beginning in the 50s or 60s with no recovery. It's shown that there's a degeneration of dopamine-releasing neurons. Dopamine normally prevents excessive activity in the motor centers of the brain, and usually there's the symptoms of involuntary muscle contraction, such as a pill rolling motion with your fingers. People can become more clumsy, have illegible handwriting, and slower gait. There's drugs and physical therapy for treatment, and surgical techniques to help relieve tumors.